Hello, good morning, everybody. And thank you all for having me. And thank you in particular to Masood for putting on this exciting conference. I only wish I could be there. I was really looking forward to it. Let me just share screens here and we'll get started. So I'm going to be talking to you all today about genetic insights that we get from animal models of migraine. And I have a couple disclosures. They're basically grant funding, except for one advisory board that is unrelated. So why? In particular, why genetic models? Why do we spend so much of our time using genes from rare family variants of monogenic migraine put into mice to study this disease? Why would we want to do that? And the primary answer is that these models offer just exquisite mechanistic precision. If you have a gene that codes for a protein and it's mutated, you know that that piece of molecular machinery is the start of your understanding of the disease. You can't get that with almost any other technique. And then, of course, if you know a piece of molecular machinery, the other way to define that is that that's a molecular target, which is, of course, a really great way to find drugs. So that's really the main reason, is we can just get incredibly specific with these genetic models. But all that specificity, what use is it if this is only relevant to a form of monogenic migraine that only affects a few dozen people on the planet? You could be all specific, but what does that get you? How does that affect the rest of the people with migraine? Well, signal from the most recent genome-wide association study for migraine, studying not monogenic risk, but polygenic risk, the risk in the population, suggests that these studies could be useful. So circled here under this Manhattan plot is CACNA1A. That is the gene that is mutated in familial hemiplegic migraine type 1. It's a PQ calcium channel. But this data has absolutely nothing to do with that. This data is single nucleotide polymorphisms in that gene, variants in that gene across a population of over 100,000 migraineurs compared to nearly 800,000 controls. So this is definitely population relevant, and this gene is coming out as highly significant in this assessment of polygenic risk. So these monogenic variants may actually be relevant to the population. Moreover, and I'm going to talk about this part of things, I'm certainly not a GWAS expert, is that fundamental themes emerge from rare models. And this is true across diseases. We're not the only folks who use genetic models to study disease. This is a relevant tool across biology. So we're going to be talking about monogenic models of migraine with aura. A third of migraineurs, we're not talking about migraine without aura. We will, I'm sure, get there. But right now, migraine with aura gives us more purchase, more models, more specificity. And if we focus on this top box here, there are several brain cells that are loci of known mutations in migraine. The first, FHM1, is the one I just described. A gain of function in a PQ calcium channel in excitatory neurons. There's a mutation in FHM3 in interneurons and inhibitory cells. There's a mutation in FHM2, familial hemiplegic migraine 2, which we're going to talk about, that is actually confined to astrocytes. And finally, there's another migraine mutation that we'll talk about in the casein kinase 1 delta gene, whose cellular locus is unknown. It may be in multiple cells. We'll show evidence that it is active in neurons. Now, all of these mutations, very different as they are starting to generate common themes. So this is basically showing some of the mutations localized on the brain and some of the symptoms or some of the phenotypes that have been gleaned in animal models of disease. And I'm not going to go through all of these, but several themes come together. The first is of sensory amplification. photophobia and gains in somatosensation or pain sensation, susceptibility to spreading depolarization or cortical spreading depression, which is, of course, the mechanism of migraine aura, which gives you faith in that these are models of migraine aura. And also, we'll talk more about this, disrupted glutamate homeostasis. So we'll start, well, I'm going to tell you two stories from two wonderful grad students who are now postdocs. First is Pat Parker. He is now a postdoc at Johns Hopkins, and he studied the FHM2 mouse, and we'll get into it here. So FHM2, the mutation we study, the mice we study, have a gene that comes from a family in Italy, and this is a loss of function in the sodium potassium ATPase, a specific form that's specific to astrocytes. And they have hemiplegic migraine with aura. As you might expect, they have a decreased threshold for spreading depolarizations. And we think that this is due to slowed reuptake, which is one of the functions of astrocytes. They reuptake potassium and glutamate, slowed reuptake in the astrocytes. Why would this be the case? Well, we know that the sodium potassium ATPase, this is extracellular for an astrocyte, intracellular. The ATPase pumps sodium out and potassium in. And that obviously brings potassium reuptake into the equation. But the ATPase also basically powers all of the other pumps in the cell. And one of those is GLT1A, the glutamate transporter. So the glutamate transporter uses the membrane gradient generated by the ATPase to uptake glutamate. So if you have a dysfunction in the ATPase, you have a dysfunction in glutamate transport. What we wanted to know, this was all in vitro data done by our colleague Daniela Pietrobon. We wanted to know if this is relevant in vivo. Is glutamate uptake messed up in these animals? And so here we have our mouse colleague. This is a male mouse here, and what the mouse is doing is they're running on a ball. They get used to this. They have a little helmet, and that helmet fixes them under the stage of a 2-photon microscope. You can see he's whisking. You see that little light blinks up there, and that's used to give a whisker puff, and then we can record in the brain what the whisker sensory response is. And we used a number of different imaging techniques as well as indicators of glutamate and of calcium. So in this case, what we're showing is glutamate imaging, very, very fast imaging, and we could get basically in response to the air puff, a glutamate transient, and we could measure the decay of that transient. So we could measure the reuptake. And what we found is that to both of the time constants of this decay, they were slower in the FHM2 animals. So we basically confirmed that there's impaired glutamate uptake in these animals in vivo while they're walking around getting puffed in the face. So much for that, and that's great. But where we really got excited is we started seeing these strange things, these what we call glutamate plumes, which are little puffs of glutamate that are focal, concentric, very high amplitude, but rather small, about 20 microns in size, smaller than a lot of cells, and not in a layer that contains cells. They were completely distinct from the sensory activity that I just showed you. They had nothing to do with it. When you puffed on the air puff, the plumes didn't care. And they were much more common in FHM2 animals. And so we concluded we were looking at a previously unknown form of glutamate signaling. To make a very long story short, we were studying an astrocyte mutation, so we thought, okay, well, this must originate completely in the astrocyte. And while inefficient glutamate clearance from the astrocyte was very important, anything else we did, we could not generate plumes by manipulating astrocytes. What we found was that the presynapse of the neuron was actually incredibly important. So it was glutamate released from the synaptic cleft that was the required source of glutamate. So to put it another way, the plumes occur when neuronal glutamate release is not met by sufficient glutamate uptake from the astrocyte. It's a folie à deux between the neuron and the astrocyte. Well, what does that have to do with migraine? Is this just an artifact or sort of epiphenomenon? This data started to convince us that it wasn't. And basically that is that plumes predicted the onset of spreading depolarization. So I'm going to play this movie here. You'll see raw data and then filtered data on the right. We're increasing the concentration of veratridine, which induces SD. And what you can see is as we hit 0.75 millimolar, you see this. I'll show it again. Nothing much happening. And then at around 0.5, you'll see some plumes popping up, a few here, and then suddenly a bunch of them, they become confluent, and that becomes the nidus, the focus for the CSD wave. So these plumes are at least a biomarker of SD onset. But then when Pat used nickel to inhibit the plumes, he found that you could actually also suppress SD, i.e. increase its threshold significantly. So not only are they a marker of SD onset, they are a mechanism of SD onset. Moreover, plumes occur during the SD wave. Here are constellations of plumes going off as the wave passes. And here you see them on the SD glutamate transient, these little ticks. So they're involved in SD, and this is regardless of genotype, not just in the FHM2 animals. Thus, we concluded they're potentially relevant to not just migraine, but other diseases where SD occurs, because SD is not just a migraine phenomenon. So in summary here, we have an in vivo mechanism of action of the FHM2 mutation, slowed glutamate uptake to sensory stimulation in the awake animals, and then these weird non-canonical glutamate release events. They're common in FHM2, rare in wild type, so a mechanism of the FHM2 phenotype. They're gated by astrocyte glutamate transport, but they depend on neuronal glutamate release. They contribute to the onset of SD, and they occur during SD. So, and this is in wild type and FHM2 animals. So we're discovering a mechanism of the FHM2 mouse, but we're also discovering a mechanism that's relevant across potentially any disorder where SD occurs. All right, second story, the CK1D mouse, and this is the work of Pratyush Suryavanshi. And this is the mutation that I told you where we know the molecular mechanism. It's a mutation in this casein kinase, which is a very ubiquitous protein. But unlike, say, the FHM2 mutation, where we know that this is expressed in adults almost only in astrocytes, casein kinase 1 delta is all over the body. And so we're still trying to figure out where it might be exerting its effects. And Pratyush focused on the neurons. So before we start, we know that there are phenotypes in humans with migraine. And one of these is a deficit in sensory adaptation in the interictal phase. So if you send in a visual evoked potential or a somatosensory evoked potential in a person without migraine, the repeat stimulus will decrement. This is a known feature of sensory networks. In a person with migraine, it'll decrement more slowly. And this is something that occurs over the migraine cycle. So normal habituation or adaptation in a healthy subject, lack of habituation in episodic interictal migraine that changes during ictus, but then the phenotype gets even stronger with chronification of migraine and medication overuse headache. So what about this mouse? This mouse, we had a role in discovering it a few years back. There were mutations found in two families with familial migraine and advanced sleep phase. And the sleep phase was the rare phenotype that allowed us to pick out the needle in the haystack, this mutation. Mice expressing this gene, this mutation, had increased migraine relevant nociception. They responded more to nitroglycerin. But more relevant to our inquiry here, they had an increased susceptibility to cortical spreading depression or spreading depolarization, the mechanism of the migraine aura. And Pratyush wanted to study the neuronal responses that might account for this SD susceptibility. And what he did was use a brain slice preparation, and he looked in sensory cortex of CK1D mice. And what he did was a recording paradigm where he records in a pyramidal cell, an excitatory neuron, and stimulates from lower down in the circuit. And he records both excitatory and inhibitory inputs to that neuron using patch clamp technique, which allows you to basically dissect excitatory postsynaptic currents from inhibitory with a combination of pharmacology and of holding the membrane potential at different levels. You can dissect inhibition out from excitation. And what he found quite quickly with these animals, or the brain slices from these animals, is as he stimulated this circuit at a given frequency, he saw no difference. CK1D in red, wild type in blue. But as he increased the frequency of stimulation, he saw a deficit in the adaptation or habituation of that synaptic response. And this became highly significant at 50 hertz at higher frequency stimulation. So a frequency-dependent adaptation failure at excitatory synapses, apparently recapitulating in microcosm what we see phenotypically in migraine patients. Now, in microcosm, because we can actually rely on years of work in synaptic biology, we can actually be quite precise about what causes that adaptation failure. Not to belabor too much, but the presynapse, the presynaptic terminal has a readily releasable pool and a reserve pool here in green and white respectively. And you can actually quantify through these stimulation paradigms the parameters of these pools. Basically, if you basically quantify these by adding them up, making a cumulative amplitude of all the excitatory postsynaptic currents, the steady state slope gives you the replenishment rate of the synaptic vesicle pool, and the y-intercept gives you its size. And what Pratouche found is that the y-intercepts were significantly larger at 20 hertz and 50 hertz, suggesting that the adaptation deficit in the CK1D mice was due to a larger readily releasable pool in these animals. So again, what does this have to do with the migraine phenotype? In this case, the susceptibility to spreading to polarizations. Well, we know that if we lower the calcium concentration at the synapse, we can inhibit vesicular release. And when Pratouche does that, he can rescue the habituation deficit that we saw in the CK1D mice and normalize the size of the readily releasable pool. And when he applies that same paradigm to SD generation, in this case in brain slices, he finds that whereas at baseline, the CK1D mice have a lower threshold for SD, a greater susceptibility, as we showed before in vivo, when he reduces presynaptic calcium, he rescues that SD phenotype. So once again, we have a phenotype that we found in this genetic model that we can show is relevant to the core phenotype of the disease of migraine with aura, which is SD. So in summary here, CK1D neurons have higher steady state currents, so decreased adaptation in excitatory but not inhibitory neurons. I didn't tell you about the lack in inhibitory neurons. The mechanism is a larger releasable pool, readily releasable pool of synaptic vesicles. And the conditions that rescue the readily releasable pool deficit also normalize the SD susceptibility in this model. All right, bringing this back to the original question, why do we study these things? What are all these details that I told you that I'm excited about? Why should you be excited about them at all? Well, the reason is the common themes, right? So in the FHM2 mice, there's this folie à deux between neurons and astrocytes. The result is increased green dots in the presynaptic cleft, increased glutamate in the presynaptic cleft. This fiddly adaptation phenotype in the CK1 delta mice, what's the net result? Increased green dots in the synaptic cleft, more glutamate in this, more release of glutamate in two completely different models of disease from completely different families with completely different mutations. And it's not just these two models. If you go back to the original model we mentioned right at the beginning of the talk, the first mouse model of migraine, the FHM1 mouse, this is a gain of function of a presynaptic calcium channel, resulting in more glutamate release in the synaptic cleft. So 3 completely different models of disease, all saying the same thing about migraine with aura, while the vagaries of one mutation or another might be unique to the dozens or hundreds of family members that are affected, the common theme is likely relevant well beyond those small numbers of people. And that's why my lab at least uses these models. So I just wanted to thank Pat and Pratouche, and also Daniela, our wonderful friend and collaborator, and also everyone else in the lab. And thank you all very much for listening. And I would say I'll take questions, but no questions in this session. Thank you all for listening.